So as we reconvene, I'd like to take this moment to make some more appreciations. We need a big round of applause for Luke McMahon in the booth, and I know you can hear me, because he's been... We had a little excitement this morning, and he helped me to breathe, so that was great. I also would like to thank the committee members. I know not everybody's here. If you're on the committee, just give a wave, okay? There we go. Great. And you'll see from the back of your program what a wonderful and diverse committee that we've had. Excuse me. And if you are a committee member or a speaker and you haven't received your modest thank you gift that looks like this, come see me. I would like you to have it. And finally, I just want to say a couple of words about our hosting, our primary hosting organization, which is Planet Connect. Planet Connect has been in business for about 20 years. It's an outgrowth of the founder's work at Bell Labs, where an engineer 20 years ago said, we need to learn about the internet, called a librarian and said, help me put a program together. And one thing led to another. So what Planet Connect does is for typically the big pharma, but also some ag science companies, does internal, proprietary, secret meetings when they want to bring together 1,000 of their, I'm looking at you, glad you're here, or I say 1,000 of their researchers, for example. And so Planet Connect does all the logistics and all the back end so that the content can flow beautifully. So about 5 years ago, the Biopharma Research Council was started so that we are not secret, we are ecumenical, we want everybody to know what we're doing, and we like to include everyone in our process. So it's a treat for me. In February I'll be here for 2 years. It's a really wonderful organization, and as I like to say, we're very flexible. If you and I think it's a good idea, we can make it happen. I don't have a whole lot of bureaucracy I have to deal with. If we can make it work, we can just do it. So don't feel shy about saying, you know what, I think we should, and maybe we can make it happen. We do webinars, a lot of webinars throughout the year, and that's often a great way to stay in touch and for us to provide you with some profile to talk about your organization and what you're doing, as well as the activities like today, like the big conference we do called D2D, Data to Drugs and Diagnostics, which will be about this time next year in the corridor between Philadelphia and New York. This March, outside of Princeton, we're going to be doing a really cool 3-day retreat called Best Practices in Outsourcing, which is looking at preclinical and early clinical. And it's a really executive level program looking at how do you build the relationships, actually some of the kinds of things we've been talking about today. So with that, I will hand things over to Deborah Thompson. Thank you very much.
Good afternoon. It's a real pleasure to be back here at the Biotech Center and to work with such an awesome group of breakthrough technologists. What I'd like to do is sort of follow on from what we learned a little bit before lunch. Before lunch, we were talking about partnerships and how do you develop partnerships that will lead to the kind of breakthrough technologies that we're going to hear about this afternoon. And so what we have is a panel of 4. We have our 2 professors, Dr. Rodolphe Barangou from NC State, Dr. Katerina Galippi from UNC Chapel Hill. We have someone who's more at the interface between university and industry, Dr. Asha Krishnamurthy. I'm sorry, I practiced it and everything. And then, of course, somebody from the big bad industry side, Dr. Andy Murphy. And I will let them talk about their work and remind them that one of our roles here is let's try to drill down into some of the specifics in this panel. And we'll start with Rodolphe, who tells me that this is much too exciting for him to actually sit down and talk about. So he's going to come up to the podium.
Thank you. Good afternoon, everyone. I think it is indeed, arguably, much too too exciting to sit down and talk about CRISPR. We actually have a number of CRISPR experts in the room. We have some technical experts. We have some tech transfer office experts on CRISPR. We have some IP experts on CRISPR as well. But we may have some people in the audience that haven't heard about CRISPR yet, though it's ever so hard to imagine. So I'm going to try to do today is walk you through very quickly in 6-7 minutes, why it's a very hot topic, why now is the time and the window to get into the CRISPR game is still open, but it's been open for a while now. Why Carolina is the place, and there's some tremendous business opportunities across different types of businesses that CRISPR holds tremendous value for. So in a nutshell, CRISPR is a novel genome editing and genetic engineering technology. You can see CRISPR as a molecular scalpel which has the ability to precisely, specifically, transferably, and efficiently, and affordably, and quickly target any sequence of interest in any cell and edit it in any way. So you just have a way to come in, cut in very precisely, and then change the sequence of your DNA at that exact precise location. There's many advantages, again, programmable, specific, transferable, efficient, precise, affordable, quick, easy to use, multiplexable, and the like. And all those are the reasons why CRISPR has become nearly ubiquitous for genome editing already. But notwithstanding this, it's a very young technology. It's only been on the market for about 18 months. And like most technologies, it needs to be improved, and we have to be somewhat patient. What's really interesting is that it has already been used for many different applications in the ag space, in the biotech space, and in the medical and translational medicine space. And you can use CRISPR and different versions of Cas9 for genome editing and or translational control and or site modification with epigenetics. So you don't have to take my word for it. This is a graph of the actual CRISPR publication and literature path. If you go back just one PhD cycle ago, there was about one paper a month. Then within a couple of years, one paper a week, and now we're at above one paper a day. And beyond this, CRISPR won last year Discovery of the Year in Nature. It was #1 runner-up in Science. And whether you're Science or Nature or Cell and Molecular Cell, CRISPR is everywhere, almost every week. You can get your CRISPR news on Twitter. It has taken over the scientific world. What's interesting is that beyond the scientific world, CRISPR has already made it to the general public. And if you look at the New York Times, look at NPR, you look at Twitter, you look at YouTube, you look at The Economist or Time Magazine or Forbes, CRISPR is everywhere. And I think this speaks volumes with regards to the pace at which things are changing and how much promise has already been leveraged in the real world. And most importantly, beyond the scientific part and beyond the public visibility part, what's really amazing is the pace at which CRISPR has taken over the commercial world, again, across ag and biotech and medicine. And interestingly, across the board in terms of types of people that are harnessing CRISPR technology. We have Fortune 50 companies, people like GE or LifeTake or Sigma. You have mid-sized companies, you have small startups, you have VCs, and you have not-for-profits. And I can hardly think of any one technology, especially in biotech, that would have garnered that much interest in that short a timeline. So arguably a quite compelling business. So why here, why now? Why in Carolina, why NC State? Why here locally in the RTP? So we have some of the most published, most cited scientific people on campus at NC State. And there's a few labs that work on CRISPR, including my lab, the CRISPR lab. We have a very active and engaged tech transfer office. Kultaran is here. He's an expert. If you have any CRISPR IP questions, you can ask him. If you don't like his answers, you can ask me. And then there's already some ambitious plans and recognition of the value of CRISPR locally with the NC Biotech Center funding some CRISPR work, Celestial Innovation Fund, NC State funding some work. And I think this is just the beginning of a few funding cycles moving forward. Locally, there's a lot of core experts and different expert teams, people that publish in Science and Nature Biotech, Molecular Cell, and so on and so forth. And we have people on campus that have expertise across the trifecta, really the scientific underpinning of what CRISPR is and how it works, then developing next generation technologies for CRISPR-Cas systems, So turning science into a technology, and then last but not least, using that technology and apply it in the real world, in human cells, in stem cells, in plant cells, and beyond. And then last but not least, we have quite a collaborative network of heavily engaged people, including academia, nationally, internationally, including large companies and including some smaller startup companies and even governmental agencies for different purposes. Now, if we talk about the science for just a minute, things that we are working on are enhancing next generation CRISPR guide RNAs. So if you know about CRISPR, it's really optimizing guide composition, guide design, guide specificity, and guide efficiency at the location of cleavage and for targeting. And then to go together with the CRISPR guides, we're working on the Cas9 nucleases, which are the proteins that generate the double-stranded break. We're working on a whole suite of orthogonal systems with Cas9 and the guides. We're working on a panel of very diverse and very safe Cas9s. And a lot of the Cas9s currently available to the public and to companies on the market are derived from pathogenic streptococci or Neisseria and Clostridia as well. And we're working on very user-friendly, relatively safe organism-type derived Cas9s. And then there's still work that has to be done on delivery methods, safety, toxicity, and everything that's needed for enhancement in the industry. So quickly, this is a list in chronological order of how CRISPR has been used to date, originally for high-resolution bacterial genotyping for pathogens, Salmonella and E. coli and others, proprietary strains from the biotech industry, harnessing CRISPR for phage resistance, and it's been heavily used already in the food supply for the genesis of fermented dairy products like yogurt and cheese, vaccinating bacteria against the uptake of undesirable genetic material like antibiotic resistance genes, genome evolution studies for hosts and their viruses, genome editing, that goes without saying, this is what the CRISPR craze has been all about, using genome editing to generate cell line development for model disease, liver cancer, BRCA1, just two examples of many, using a deactivated version of Cas9 to do transcriptional control, either turn things on or turn things off or turn things down, imaging chromosomes, using fusion proteins between GFP and Cas9, and then gene therapy. There's a lot of work right now that has tangibly established that you can use Cas9 to correct and edit DNA sequences for Duchenne's muscular dystrophy. You can carve out HIV, cataract, tyrosinemia, beta thalassemia, cystic fibrosis, and the list goes on and on and on. There's some papers recently that have shown you can use CRISPR as next generation antimicrobials to precisely remove selectively organisms of interest and leave the rest of the population alone. Gene drives to get rid of mosquitoes that are responsible for malaria. There's some interesting conundrum there. And then last but not least, using CRISPR to simplify genomes of interest. So altogether, I hope those 5 minutes allow you to see it's a pretty hot topic. Now is the good time with the CRISPR craze. There's a great place to be in the RTP to do CRISPR research. We have some great teams and experts at NC State. There's tremendous business opportunities for ag and for biotech and for pharma research. There's tremendous deliverables we offer with the guys in the Cas9s. And then last but not least, a lot of people have already established that it's a very sound investment to invest in technology. And I hope some of you will add the CRISPR element to your research program. That's what I have, and we'll go on to the next speaker and hold questions for the end, right?
Please, please.
All right. Thank you.
Now, in the preliminary conversations that I had with both Rodolphe and with Katerina, both of them indicated how really important it was to them to have the interaction between their university-based labs and industry for an industry contribution. So I hope, Katerina, you'll have a chance to talk about a little bit of that now.
Sure. Is my microphone on? Is that working?
I don't think so.
Okay, I think this one works. Okay, great, thanks. All right, thanks, Rodolphe, for that really interesting talk. I have to admit that I was naive to CRISPR before that talk, but it sounds really exciting. And I think it's an outstanding example of how academic science can be brought out into industry. And what I hope my talk will touch on today is a little bit about how industry can be brought into academic science, so sort of the flip of that. First, let me just say that I'm a faculty member in the biomedical engineering department at UNC and NC State. It's a joint department between the two institutions, as Fran described in the last session. My laboratory home is at UNC, but my appointment is truly joint between the two universities, and I have access to all resources on both campuses, so I consider myself to be faculty at both UNC and NC State. Okay, so just quickly, a brief overview of what we do in my lab. So many of us are familiar with the concept of palpating to perform some form of clinical diagnosis. You go into the doctor and you say, my arm hurts, and your doctor will probably touch your arm and push on it a little bit to see if it feels harder or softer than it should, or if it just feels strange. What your doctor's assessing when he does that is really the mechanical property of the tissue. We can do that, but with much greater precision and objectivity and at great depth and with great focal and temporal precision using ultrasound. This is the same ultrasound scanner that you see that takes pictures of babies and does all those other great diagnostic functions. This is the same clinical diagnostic scanner. We have a research relationship with Siemens, which I'm going to talk about in a minute, that allows us to manipulate the way that this scanner performs ultrasound imaging. So we can manipulate the scanner to do things in sort of an unconventional way, and that allows us to do what I consider to be remote palpation. We use the ultrasound energy itself, but very focally, very precisely. We're talking about hundreds of microns is the sort of the cross-sectional area of the push. We can use the sound energy to push on tissue very subtly, displacing the tissue only a few microns, nothing that can be felt or sensed by the patient. But because ultrasound is so well tuned to monitoring motion, we can then survey the mechanical response of the tissue to that excitation, and that tells us about the mechanical property of the tissue. We're not imaging the acoustic property of tissue as is conventionally done in ultrasound. We're imaging the mechanical property of tissue, which is diagnostically relevant. And I'll show you a couple examples about how we use this. So through the course of developing this technology in my laboratory, and there are many different applications for it, both preclinical and clinical, both from sort of a diagnostic perspective and a basic science perspective, we've had a variety of different industry partners, but the three that have had the most significant impact have been Siemens Medical, Marine Polymer Technologies, which is a small pharmaceutical company in Boston, and Novo Nordisk. And I'm going to talk about in greater detail, for the sake of time, only the relationships with Siemens and Novo Nordisk. So first, Siemens. This is a win-win relationship for both us and Siemens, as far as I'm concerned. We win proprietary access to their system architecture and control mechanisms. So that means that we can get this scanner to do things that it wouldn't otherwise be able to do. Importantly, this scanner is FDA approved. I could certainly piece together an ultrasound scanner, or I can buy a research-dedicated ultrasound scanner that does not have FDA approval. But then when I try to translate that to clinical studies, the IRB gets a little upset. So having a scanner that is approved for use by the FDA, that we know is safe in terms of its energy outputs, is very important to my work in terms of translating this technology into real clinical studies. And I'll show you some clinical data in our analysis in this talk here. So we have access to the system, and moreover, we parlay with the engineering team at Siemens. So if we have a question about how the system works, or if we have ideas on how to make it better for our purposes, we can talk with the engineering team there, and we have a nice relationship with them. What do they win? Well, they get access to what we do, and they know it before anybody else does. So as we're developing new clinically relevant technologies, they are aware of what we're doing. They are surveying the clinical relevance of what we're doing. And of course, we're sort of testing that out for them because we're publishing papers, we're getting grant support, et cetera. So they have a feel for what the general imaging community is interested in. Moreover, we collaborate with lots and lots and lots of people. Many of them are not engineers. So we work with a lot of physicians, we work with biostatisticians, we work with people outside of their immediate universe that bring to the table some unique perspective that's valuable to them. And importantly, they get access to talent. So when my graduate students leave my lab, they are trained to work on Siemens systems. There is no learning curve for them. If Siemens chooses to hire them, they're ready to hit the ground running when they get into their jobs. Okay, So one of the projects we're working on in collaboration with Siemens is characterizing the structure and composition of atherosclerotic plaque. So hopefully nobody in this room has atherosclerotic plaque, but if you do, you might have considered that there are different types and varieties of plaque. Some of them are more dangerous than others. And one of the key features that makes a plaque really dangerous, and by dangerous I mean likely to cause a heart attack or stroke, is that it has a soft center. This is generally composed of some sort of lipid material. It's covered by a stiffer fibrous cap. Without RFI, this technology that I'm showing you here, which stands for acoustic radiation force impulse ultrasound, without RFI, this plaque just looks like a white structure on the B-mode image. You can tell that it's there, but you can't see anything about its structure or composition. With our technique, we can tell you that this plaque has a soft center and therefore is a dangerous plaque and needs more critical clinical attention than a plaque that's primarily fibrotic or primarily composed of calcium, for example. Here's the histology that validates that. These are specimens that were taken from carotid endarterectomy patients. I'm just showing you one example here. But basically, these patients are having their carotid plaques removed. We image them immediately prior to surgery. We harvest the tissue and do the matched immunohistochemistry for validation. And you can see in this case that there's a necrotic core here and an area of inflammation and a calcium deposit. And those features were all apparent in our RFI image, but would not be apparent on a conventional B mode. Here's another clinical study that we're doing with Siemens. This is clinical diagnostic for muscular dystrophy. We're trying to monitor dystrophic muscle degeneration in boys who have Duchenne muscular dystrophy. So many of us know that Duchenne involves a genetic mutation, and it typically presents with physical impairment around age 5 and rapidly progresses to the point of by age 10, boys are generally unable to walk. And by their late teens and early 20s, they usually die from cardiac and respiratory failure. So this is a really debilitating disease. We are working to develop a non-invasive biomarker that can be used to validate outcomes from different treatment trials. So there are a variety of treatments that are available for Duchenne on an experimental basis. Many of them have not been approved for clinical trials because there is not a validated outcome measure. We're trying to create that outcome measure. You can see here, this is a 5-year-old boy, so early in the presentation of the disease, versus a 9-year-old boy, so later. This is pretty close to when this boy is about to lose ambulation. Here you see the muscles. This is the rectus femoris in cross-section and longitudinally in the standard B-mode image, and there's not a whole lot of information about the composition of that muscle. But when we turn on our imaging technique, the red here is indicating necrosis and inflammation. And you can see that the 9-year-old has a much higher percentage of necrosis and inflammation than the 5-year-old. And we do this serially in boys, so we monitor their progression toward inflammation and necrosis, and similarly, their muscles turning fibrotic over time. We do a corollary study in preclinical Duchenne muscular dystrophy. This is a golden retriever model of the disease. And we see on the top slides here, you're seeing a normal dog. On the bottom slides, you're seeing a dog that has Duchenne, a version of Duchenne, a corollary of Duchenne. You can see that in the 3-month time point, there's a lot of inflammation and necrosis present in the Duchenne dog, but not in the normal. And that is being replaced by fibrotic tissue over time in the Duchenne. And this is the histology that confirms what I just said. Okay, so our relationship with Novo Nordisk is a little different. Obviously, Novo Nordisk is not an imaging company. They're a pharmaceutical company, so they're not as interested in the diagnostic potential of the techniques that we develop. But they do make hemophilia drugs. Whoops. They make hemophilia drugs, and they're interested in knowing how their drugs impact bleeding non-invasively in vivo. So they don't want to harvest the blood and then do some kind of coagulation assay ex vivo, because bleeding dynamics alter dramatically if you take the blood out of the system, right? So they want an in vivo bleeding assay, and they asked us, can you develop this for us? So we spent quite a few years thinking about this question, and we've come up with a technique to do that. Again, we're using our fundamental basis of pushing on tissue using acoustic energy. And when we do that, if we apply some sophisticated signal processing, we can find regions of hemorrhage. So here's an example in a dog where this is a small diameter vein. This is about a 2 millimeter diameter vein in the hind limb. And we puncture it with a standardized puncture. And then we watch it bleed. And this is time after the point where we've punctured it. This is at the time of puncture, and then 10, 30, and 100 minutes after puncture. Pixels that are red here are the pixels that we've detected to be regions of hemorrhage. By looking at how this area changes over time, we can assess bleeding dynamics like rate of bleeding and time to hemostasis, which we then use to study drug response, so dose response. In this study, we were looking at normal dogs versus hemophilia A dogs treated with 3%, 10%, or 100% recombinant factor VIII replacement. And we saw a statistically significant difference between the naives and the treatment level at 100%. Similarly, we can look at bleeding rate, and we saw time to hemostasis decrease sort of systematically here with increasing dose level. So this is data that we created and gave to Novo Nordisk in response to studying their treatments. As I mentioned earlier, my research is very interdisciplinary. So I'm working with folks from cardiology, pathology, vascular surgery, neurology, nephrology, radiology, biostatistics, mathematics, and veterinary medicine. And that's just for the data that I've shown you on these slides. We have additional projects with folks who are not represented on these slides. So as we develop these industry collaborations, I want to make it clear that the collaboration is not with Katerina Galippi. The collaboration is with Katerina Galippi and her entire research resources, which include all of the people on this slide. I think it's a pretty unique opportunity within academics that we have access to so much expertise, both intellectual expertise and physical resources that I think are hard to reproduce within any one organization outside of academics. And the last point I want to make is, I'm really just a case study. My laboratory is one example of how industry-academic relationships can be symbiotic. When you look at the map here, and this is an image that I took actually from the NC Biotech Center's website, so thank you for this picture. But as you can see, in this area, there are so many resources within our state, and particularly within, a 20-mile radius of where we're sitting right now, that we can really exploit these resources, I think, to even do a better job of enhancing collaboration between academics and industry so that some of these opportunities can be exploited to their fullest potential.
Thank you. Let's move to Dr. Krishnamurthy's presentation, and this is exciting because he sits, as I mentioned, pretty much at the interface between university and industry, and his role as Deputy Director for RENCI is to manage and enhance research partnerships with both faculty and with industry that's in the area.
Thank you very much. Yeah, that seems to be working. So thanks for the opportunity to talk to all of you. My talk is going to be a little bit more broad-based than what you heard so far. Very interesting things that we heard so far. But I want to sort of give a picture of RENCI and some of the projects that we are involved in that may be of interest to you all. So I'm going to basically talk quickly about RENCI because in some ways RENCI is an unknown entity, but I think a very significant resource if you know about it. So I want to mention a little bit about that and three of the projects that we are involved in right now out of many other projects we do. So RENCI stands for the Renaissance Computing Institute, been around since 2004, and sort of I don't like the, okay, anyway. So the mission, our mission is to be a leader in cyber infrastructure research and development. Basically, our goal is really to enable faculty, industry, other researchers to be able to make use of computing technology in all of its ways, shape, and form to do their job better. So we really want to act as an enabler. And certainly in the biomedical engineering, you heard about the joint projects or the joint department between state and UNC Chapel Hill. We're going to one-up that a little bit, and you can see up there we also include Duke as part of our team of players. And it really is that way. When RENCI was set up, it is set up so that our advisory board consists of the provost and the chief research officer from all three of these institutions. So We meet on a regular basis, and they provide us direction on how to go. And we work extensively with faculty and other researchers across all 3 campuses. Okay. So basically, this wheel or this gear, as you might choose to call it, summarizes all the different things we do. Our focus of late has really been on data and analytics and high performance computing. We have always done visualization, software development, etc. But the idea is that collaborative projects are what drive Renzi. Almost every single project that we do is in collaboration with another institution. There are very few projects that we do that are completely internally Renzi focused. Okay. I'm going to skip these since I don't think these are all that important. So I want to talk about some of the projects we have going. One is called the National Consortium for Data Science, NCDS. And there's lots of interest, some would call hype, about big data and data science today. And what we believe is that underneath all of that, there is a real change happening in how many fields that have access to data and have used data always, and new fields that are now gaining access to data because of changes in technology, have to understand how to use it and exploit it better for accomplishing their mission. So we call it as our sort of tagline for this is data to discovery, which is how can you actually get insights and real actionable things from the world of data around. Okay. The main thing I wanted to point out was that the Harvard Business Review claims data scientists as the sexiest job of the 21st century, which I thought was kind of crazy, but anyway. So one of the forces driving data-centric research, if you will, is the enormous growth of data. I'm sure you're all aware of this, but this chart kind of tells you, how the explosion is happening today. Okay. So again, what is driving the data explosion? Easy access to data, more ability to compute, the sensor explosion, the so-called Internet of Things, where every single thing you own in your house and elsewhere is going to be now connected to the internet, and of course the growth in biological data, which would be of relevance to everybody here. Okay. So I'm just going to tell a little bit about what NCDS does. So it's a partnership. It's a public-private partnership, which you have heard of from others today. So we carry on that theme of actually working across industry, nonprofits, government agencies, and academia. So here is our current list of membership, as you'll see. We are, at this point in time, somewhat North Carolina focused, although we do have, for example, membership from Drexel, Deloitte Consulting, that are not necessarily based in the Triangle, but others have a strong Triangle presence, if you will. Some of the things we have done, which are more programmatic in nature, we are creating what we call the data lab and observatory. This is a place where we are reflecting data that may exist at many different places. And one of the themes that you'll see in my later thing is that the ability to share data and be able to have sufficient metadata associated with it so you can make sense out of it is, I think, one of the crying needs going forward. And so the Data Laboratory Observatory is an attempt to build tools as well as a mechanism to make it easy for researchers, first of all, to find data as well as to be able to deposit data should they so choose, or share data if they so choose. We have a number of other programs. Data Fellows is having faculty work with industry and vice versa. We have working groups where we sort of take a deep dive into a topic for a year. And then we organize various kinds of events, leadership summits, and also student career events, etc. Because for a lot of our members, access to talent is an important thing. Okay. So I'm going to skip this. Then I'm going to move on to the second thing I want to talk about, which is you heard about tracks this morning from Bradford Walters. Tell you a little bit more in specifically about the biomedical informatics aspect of tracks, because that's what I work in. TRACS, in case you don't remember from the morning, is the Clinical and Translational Science Institute at UNC Chapel Hill. The things that we do with TRACS is one of the BMI, Biomedical Informatics, is one of the components of TRACS, and we really support a number of activities. The three I want to point out is the Carolina Data Warehouse for Health, which is where all of the data from all of the patients that either outpatient or inpatient in the UNC healthcare system, it eventually comes to live there and is used for research in various ways. And I'm going to talk about two particular projects that RENCI has done around that. One is called the Secure Research Workspace. The other one is an effort called NC Genes. Okay. Just to give you some data about the Carolina Data Warehouse, it has approximately over 2 million unique patients. As you can see, there's data that goes back to July 2004. Last year, April 2014 was a big change. This year, I'm sorry, we're still in 2014. April, EPIC is the new EMR system that has been adopted. EPIC appears to be being adopted by every research healthcare system around the country, and UNC put that in there. So now EPIC data is also available through the Carolina Data Warehouse. So a lot of the research studies originate by a request of, how many samples, what's the cohort size for a particular study you want to do, and then maybe extract, go through the IRB process, extract the data, and that's how most research projects get launched. So it's a key component of the biomedical research. And Robert Bradford and Dr. Tim Carey are two of the key people that we work with in doing this. Okay. The Secure Research Workspace, as you can imagine, a lot of the data has patient identifiable information that is used for research, and there's always a need for how do you make use of this data while at the same time not just protecting the data and the institution, but also protecting the investigator, because you can have the best of intentions and sometimes your laptop gets stolen or something happens and suddenly you are responsible for a major data breach. So the Secure Research Workspace is actually a container, a research container that gets loaded with all of the software that you need to analyze the data. The data that you're requested gets provisioned into it, and then you can log on and work on that secure workspace, and all your action is captured and audited and available, so you know exactly where the data lives. So it protects, as I said, the institution, the patient, as well as the researchers themselves. So this was technology that was developed by RENCI and is now made available to TRACS. It's being used in TRACS, and we're in the process of actually expanding it beyond just medical. We started out at being the secure medical workspace, then we found out that there's a need for it in other areas such as social sciences and demography, etc. So we're expanding it. Okay. Another large project that we have going is called NC Genes, and this is actually a project in clinical genomics. RENCI itself, we don't get involved in the sequencing itself. The UNC High Throughput Sequencing Facility does the sequencing. We get involved in the analysis of the data afterwards. And so the entire pipeline for doing this analysis, all the computation, including the high performance computing, is hosted, developed, and run at RENCI. We have about 6 researchers who work with this, and Dr. Kirk Williamson is the head of the group, leads group. This is sort of an eye chart, but it kind of shows you the complexity of what NC Genes is and how the data flows across different portions of the analysis stream. So this whole workflow is instrumented and run out of RENCI. Okay. Finally, this is a little bit about biomedical data sharing again, and the main message here is that sharing is caring. It's important to be able to share data. And I2B2 and Shrine are two of the software products. They're both out of Harvard that now appear to be appear to be becoming the standard for for at least sharing of electronic health records across the country. But it's not as easy as it seems, even though this technology exists. And as a sort of a step in going in this direction, UNC, Duke, Wake Forest, and the health systems of South Carolina are involved in a project to actually be able to begin with, at least get counts for patient cohorts across these institutions, because there may be certain disease types where any one institution may not have enough people to create a meaningful study, but across institutions that may be possible. So this may be of interest for doing clinical trials, et cetera, where you want to engage and find what the region has to offer in terms of being able to do these kinds of studies. So this is in the process. Work is progressing, and we hope to have some preliminary data out of the sharing probably early first quarter next year. What is driving the need for data sharing? I think that the last line shows that, all those dollar signs, and basically all of the funding agencies, NIH, PCOR, in case you are aware of the Patient-Centered Outcomes Research Institute, as well as the National Institute of Health, are more and more requiring that major medical research projects have to be able to share data across institutions. And so funding is being directed quite heavily in that direction. And so all of the work that's going on towards data sharing is along that direction. So data sharing is good. You should always want to share data, but the money helps to do it. So that's the message there. Okay. Well, I mean, so what we are doing is basically trying to get, I don't think this much. Thank you.
Thank you, Asha. We've moved from an explosion of CRISPR publications to an explosion of data. And now we will move to Dr. Andy Murphy. Andy moved directly to industry from his postdoc at UNC Chapel Hill, starting with Liquidia, and recently taking a position as Director of Bioseparations Technology at Proteovec. Andy?
Oh, thank you. So I just want to take this opportunity to sort of introduce ourselves and tell you about how we're enabling technologies and discoveries. And our role, and what we see our role, is to really help people who discover new therapeutics and biotherapeutics particularly, help them transition in from the point of a few good initial experiments to putting it into people and seeing if it can really do what it's thought to do, can do. So in terms of our business model, we are ultimately a biomanufacturing group. But what we're trying to do is really focus on universities and small pharma and biotech and enable transitioning that first identification of a protein and biotherapeutic that can be useful and is interesting, and to do it in a really cost-effective way. And so that if we can use innovative approaches to reduce the cost, that we can sort of bring those regulated development activities in sooner, and two, sort of by increasing scale, sort of enable a much broader ability to study and characterize the drug product. So we're working in 3 areas on this. On the upstream side, in manufacturing protein, we're looking at just the bulk manufacturing and using different ways to approach that and to do it effectively. The second area we're looking at is the downstream side, which is the purification side, which bears a great deal of cost as well. And to look at that in a way that we can do that in a single pass and greatly reduce the number of steps and processes. And the last piece that we work with, and this is something we work with one of our partners, is to help find those optimal formulations where we deal with stability, aggregation issues, and concentration issues, so that at the end of the day, if you have a protein, you can go through this process and have a drug product that can be tested. And ultimately, developing a drug, it requires a lot of different expertises and a lot of different people. So we don't do it alone. We work with what we call the Southern Biologics Network, which is a clustering of different CMOs and service providers that can help our customers really do the types of studies that they need to do and get the work done. And so the last piece of our business plan is that as we grow and we have clusters of customers in sort of local places, that will open up labs at those geographic local sites and to help facilitate the development of these new proteins and drugs. So how this breaks down in a little bit greater detail in terms of what we bring to this process. In protein expression, we have a novel expression system. It's the STAVEC. It's based on a bacterial transposon. And ultimately what it allows is the long-term stable production of protein. And I'll show a graph in a second. So that you can put a very little bit of effort up front and then still collect protein over a great deal of time and generate a lot of protein in short order. And ultimately, the work there is focused on transitioning this into disposable cultureware that's modular and scalable so that we can both build up in terms of scale, but also put it into clean rooms and GMP spaces, which will ultimately be required. Our second technology, which is what happens in the lab in Raleigh, is based on protein purification. We call this our LigaTrap technology. It's based on small molecular weight affinity ligands. We design ligands for the proteins, and so then these ligands can go grab the protein out of complex mixtures. And if we do our job right and we design our processes right, these will be a single step and really speed up the rate at which we can purify the ultimate product. And the last piece in terms of protein formulation, we work with Soluble Therapeutics, which is a company based in Alabama. They've developed this HSC technology, which is an automated formulation machine. And basically it goes through and screens different buffers and surfactants automatically. And it's a really great technology to be able to go do that formulation work that traditionally requires a lot of material and a lot of time. And to go do that in a very short amount of time with very little protein. And then ultimately, that is the sort of final step in terms of developing a stable new drug. Just to show a little bit of data in terms of our expression systems, this is an example that I pulled that where our scientists expressed interferon alpha, and they developed these cell culture, and they were able to maintain expression over almost 10 months. So to think to start a cell culture and then have it continue going for 10 months is a very long time. And that allows to not only to generate a lot of protein, but to not have to worry about producing multiple lots over time. And so it's really neat, and it really will enable us to produce a great deal of protein for our customers. And so the second piece here, in terms of our downstream processes and the LigaTrap technology, we're really focused on replacing different types of affinity chromatography ligands, and particularly replacing proteins and peptides and other complicated biomolecules used in affinity chromatography, and using chemically synthesized ligands that we can both design and put a lot of chemical diversity into, but also create a lot of stability, both towards different types of chemistries, but also different enzymes. And this allows us hopefully to generate resins that are stable and can be reused. And we don't have to worry about some of the drawbacks of traditional affinity chromatography. And so in terms of where we started in looking at developing an affinity resin business was to look at the different types of biotherapeutics that are out there. And when you look at that, most of the biotherapeutics are antibodies right now, and they're in large volume. And the primary way to purify those proteins is to use protein A, which is a bacterial-derived protein, which is then put on a resin. And it works well, but it costs a lot. It's not chemically and enzymatically stable. And in some cases, it just doesn't work. And so there's a lot of issues in terms of how harsh you have to treat the proteins at the end to get them off the resin and get them pure. So that causes aggregation and limits its ability to be used for everything. But also in terms of looking at replacing that, we want to increase the scope, and we want to generally build tools that our researchers will be able to use. And so I I'm not going to use the laser pointer because I don't want to. I have a little bit of data here in terms of comparing our protein A versus our early prototyping data, and this is early prototyping data. And basically, we're pulling those antibodies out of cell culture media, out of great mixtures of protein, the proteins in the media, and getting our antibody back out in a pure state. And that's the protein A, that E column, versus our right prototyping column. So where this all sort of converges again is that as we're developing upstream processes, we're developing novel downstream processes. And hopefully, as our customers' processes grow, we'll be able to use our downstream technologies together and bring them together. And so when we bring them together, as those traverse into clinical trials and sort of phase 2 and 3, that we'll have those processes there at the end, and that we won't have to do any change of processes at the end. That being said, at the same time, these resins are useful tools, and that we're going to start out by providing them as analytical tools, but they can also start to displace existing processes. So just as a last slide, just to give you an idea of the diversity of different companies we work with and sort of the spectrum of skills that we bring in. We're working with people that span early target validation through phase 1 and 2 safety studies. And that ultimately, altogether, we're hoping that we can really help the small researcher bring their new drugs into clinical evaluation. So that's all I have.
what you're all engaged in and the evolution of this region as a center as a place where partnerships are playing a big role in development is really obvious. But I'm curious if, we've got university representatives here, we have business representatives here, and people who work with those institutions. But I look at the agenda and I don't see anyone from government, either local, regional government, or state government. And I'm wondering, I'd like to hear your perspectives on what, if anything, you think the role of government can or should be in systematizing this process, or making it work better, or facilitating partnerships, or should they just stay out of the way?
So, to paraphrase the question, what is the role of governments in developing and enhancing partnerships between university and industry?
So if you look at the SBIR and STTR funding framework, I would say, they are documented means to get government funding to push the agenda forward. It's unfortunate that they're not here today, obviously, so they can defend themselves firsthand.
I was thinking more of local and regional and state governments, because the federal governments provide a lot of funding. That's part of the resource kit that everybody looks to do some of the seed work and basic research. Other levels.
So for us, since we're a relatively small company, we're actually taking advantage of what NC State provides in terms of incubator space. So we're actually located on NC State campus right now. That actually provides us, and those resources provide us a great deal of things that we just wouldn't have access to in terms of like library and also facilities, and be co-locating with other scientists and potential customers. So from the standpoint of, NC State being a state-run organization, that they are providing space for us to, and funding for us to do that.
And for full disclosure, we have neighboring labs,
Yeah, we do.
which we didn't know until now.
That's good.
So I'm going to jump in and put on my hat of my previous job, which was here at the Biotech Center. Is there anybody else here right now that? So I'm going to jump in and say that. And I worked here at the Biotech Center for 3 years as a program manager, so I'm going into the past here. But look to the Biotech Center for some state support, because although the Biotech Center is not a state-run group, they are to a large extent, like 90+%, currently funded by the state. And they provide support for some of these collaborative researchers. Look to the collaborative funding grant as a particular example. Rodolphe, did you receive a CFG?
Actually, I did, and it was on slide number 5 in my talk. So I am getting funding, for my CRISPR research from the NC Biotech Center. I'm receiving startup fund from NC State University. I'm receiving Chancellor Innovation Funds, which we heard about from earlier today. So there are means, and tangible ways in which they are directly financing innovation and breakthrough technologies.
So if you're looking more at, I think your question was more about state funding opportunities or state support. That's one of a variety of mechanisms.
But, for example, the state biofuels facility up in Oxford, I think it's essentially gone, isn't it? I mean, is that the trend, or is that just an anomaly, or, I'm an engineer, I'm a consulting engineer helping with finding sites for high-tech projects, and across the board, the state cuts have been dramatic and been very difficult in terms of all the great opportunities that the state has and has seen sidetracked to some extent. And from your folks' perspective, are you seeing it? I mean, to touch on the latest question up on the front row there, I see it in my business. Do you see it in yours?
For example, this is relating to universities. So the UNCGA, which is the General Administration for all of the UNC universities, has a strategic research plan, and right now, just about a month ago, I believe that it has been funded to the extent of $3 million for about 6 research areas. So the state has indeed put some money into it, perhaps not as much as we would like. And there is a call for proposals that is going around right now where you can actually apply for what are essentially continuing funds for specific research areas and research projects. I'm aware of that because one of the areas that they have highlighted as data science, and so we are in the process of putting an application in that area. And again, one can argue whether it is too much or too little, but at least there is some movement towards funding strategic collaborative research areas.
From the academic perspective, there has definitely been a significant reduction in the amount of both federal and state funding that's available for academic research, and that has a tremendous impact on the ability to innovate. If we don't have the dollars, we can't do the experiments, we can't make the technology, that technology doesn't get to market, and it's that simple.
Yeah, go ahead.
I had a question about government on a different basis. When you're talking about sharing big data, what kind of things do you have to deal with when it comes to medical data and trying to organize these networks? How do you comply with all the privacy around medical records and also informed consent if you're talking about trying to join institutions for clinical studies and things like that?
Yeah, and those are critical and important questions that are in the process of being solved. I think that the example study that I talked about right now, all we are doing is essentially what is called getting cohort counts, which is to say, if we pool the patient information from all of these institutions for a particular study you want to conduct, how many people are there? The next step would then be, going back and being able to reach the patients and get their consent to participate in studies, etc. And of course, that's where you want to go. That's where you're starting there. But the first step we have taken is simply to be able to identify counts to see if certain studies are viable or not to begin with. As you go into how do you actually do the studies themselves and be able to engage with the patients, so that's where privacy and confidentiality comes into play. There's also issues with the healthcare systems are all very competitive, and they are also quite reluctant to share information about patients between each other. So in some ways, either you have to have some sort of an honest broker mechanism, somebody that can win the trust of all the parties to say they're not going to necessarily bias things one way or the other. And also to be able to ensure that when these things are brought together, that there's an attempt to keep things secure. That's where things like the secure research workspace I talked about comes into place, where you try not to, or you don't really allow any sensitive information to be able to leak out from the places where the work is actually done. if you will. So I don't know that all the issues are resolved, but I think everybody's aware of it and moving in that direction.
Katerina, a lot of your work is done with the canine model, but at least some of it with a human population. Do you run into the same questions?
Right. So we have to be really careful about PHI, and it's a big challenge. We have extensive training. As faculty members, we're required to go through a lot of training before we are even given authorization to perform a clinical study at UNC in the hospital system there. But it's we have to be really careful.
What is the current off-target efficiency for the CRISPR-Cas9 system, and how do you mitigate that?
So can you
Yeah,
paraphrase the
So
question?
the question is, what is the level of concern with off-target activity of Cas9, and then how is it being mitigated? 3 points. First one is a lot of the early reports about high level of off-target cleavage were overclaimed. Secondly, you can use a dual nickase to alleviate the challenges inherent to standard off-target cleavage. But third, and most importantly, if you look at the body of work and you look at the development of next generation guide, and the guide design aspect is critical. This is what early on, first generation tools to pick your target were very suboptimal. So we can discuss details of the break. Those issues have been addressed significantly to date, and there's no concerns whatsoever.
One more.
Please.
How do you compare the specificity and efficiency of CRISPR-Cas with Canon's and meganucleases?
There's a reason.
So the question?
Yeah, so the question is, how does Cas9-based targeting compare to ZFN's, talons, and meganucleases? If you look at the data and the trends, I think the results speak for themselves.
And the next
And
question is how often you get asked that question.
all the time, every day. So I think in the end, you can look at the science all you want, you can look at the users all you want. If you look at the business excitement, and the fact that it's across the board, even from companies that have a vested interest in pushing the incumbent technologies, I think the data speaks for itself.
Andy?
But how do you convince a company who, let's say, has the choice, CRISPR-Cas or other genome editing tools, to create a product, let's say, a plant, that they have also FTO, freedom to operate with this? The IP situation around CRISPR-Cas seems to be very complicated, but it's very clear on meganucleases, on canine.
And so if you'll paraphrase it, and then actually this will have relevance, this question has relevance to all four panelists, so just insert your favorite technology into the question and answer it, but we'll start with Rodolphe.
Yeah, so the question is, how is the IP landscape challenge into play? And then how would you convince, or how would a company make a decision to harness CRISPR instead of what it has freedom to operate in? For the first question, inquiring about the complexity of the CRISPR IP landscape, any adjective you can use is going to underdo it. So it's more complicated, it's more fuzzy, it's more unpredictable, it's more challenging than anybody is willing to acknowledge, to the best of my knowledge. So it is complicated, it's going to be complicated for a while, it's going to be unpredictable for a while. There's only so much everybody knows, there's what people don't know that has been filed. And notwithstanding the clear instructions of the USPTO and EPO and others globally, it is unpredictable in terms of timeline and outcome. So it's a challenge, the number one challenge in my mind. But number two, from a technical standpoint, it's obvious it is the best technology by a mile, and that everybody who has a vested interest in genome editing and beyond, CRISPR is king, and the CRISPR craze will live for a while, in my opinion, and that of thousands of others.
So if each of our panelists will sort of talk about it in your own sector, in your own research.
Well, I think in terms of my experiences, in industry is that a lot of times you have neat technologies, but ultimately if you want to convince someone to use it and change, you have to solve problems that they can't solve for themselves, or you have to do it a lot cheaper. And usually the cheaper doesn't do it quite as much, but in terms of more established technologies. But really, solving problems that people can't solve on their own is very clearly a way to get people interested in and get them to use those new technologies. And it also helps if you have new technologies that can solve problems and a clear IP pathway. It will generate the interest that's required and the momentum to get things to change.
So from my perspective, IP is an important topic because we are generating IP and yet we're partnering with a company that would benefit from our IP. So, and in some ways they're funding, maybe not in monetary dollars, but they're giving us equipment and technical support and so on to make that IP generation possible. So we have an agreement that's put in place up front before any projects are started, as was discussed in the last panel. And the agreement involves a right of first refusal for Siemens. It involves some review rights before we publish anything or apply for grant applications, but those are all within confines or limitations that I think are acceptable from an academic perspective. And there are some real benefits to having that relationship from an IP perspective too. One of them being that I get inside information about what's the most interesting technology from a commercial perspective. And the other is that if Siemens is interested in some of our IP, they pay for the patents, which when you're working in a university environment, it can often be difficult to convince your university to pay for your patents. You may believe that this is the next big thing, but unless you can convince the appropriate people in the tech transfer office that they need to put forth the dollars, it may just not be possible to pay for the patents. So again, it's a symbiotic relationship, but it's one that you have to sort of carefully tread, both for the sake of your IP and for the sake of your scientific relationship. Because if you get to the point where you don't trust each other or you don't feel like the other really is watching the other's back, then the scientific relationship degrades as well.
Yeah, that relationship of trust is really important. Asha?
I think that with Renzi, a lot of the IP we produce is in the form of software products. And we have consistently adopted a policy that to the extent possible, we will use only open source components to build our software products. And at the end, any new software products that we create as a result of putting things together or adding to it, et cetera, we will put it back into the open source. So a lot of what we do is open source software development, open source software work. Nonetheless, even though it may be open source software, we still engage with companies because sometimes they need additional help above and beyond. Open source exists. That doesn't mean that a given company or a lab has the ability to download the software and install it and configure it to be used efficiently. So we sometimes get into service engagements with them where we will help them with customizing the software for their own particular environment, for example. But where IP issues come up, we typically try to do things in a pre-competitive way where things are shared across all of our partners. But the vast majority of what we do is really open source software, I would say.
I think there was a question. Did you have a question earlier?
No, well, it was just a little bit about CRISPR-Cas. I mean, how much of the furor over CRISPR-Cas is just because it's the most easy technology to use compared to Talon's meganuclease and zinc finger?
So the question is,
as far as the versatility, I don't understand how it's different. It's the ease of use that seems to be the key there.
it's one of the, so the question is, is what percentage of the CRISPR craze is inherent to the ease of implementation?
Yeah, exactly.
So it is a driver of the rate of adoption, for sure. And the joke is that, any grad student, maybe even a UNC or Duke, can use it and they come in. And you can adapt that joke to whomever you go talk to. So I think ease of use is one thing, but accessibility, breadth of targeting, programmability, multiplexing, cost, flexibility beyond just nicking or cleavage, but in terms of targeting for repression or activation or imaging or methylation or fusions of Cas9s, and even delivery. It's across the board. And then again, it's 18 months old. There's 1,092 papers as of this morning, and it's just the beginning of the craze, and it's not abating anytime soon. And convenience is not the only reason whatsoever. And which is interesting is that despite the obvious IP challenges, again, many of the key players, if not all, in biotech, in ag, and in medicine, are in the game. So it's not just an accessibility issue, it's really the harnessing potential of the technology moving forward, for sure.
So I want to take this opportunity to thank our panelists once again, who took us from before lunch, where we talked about developing and building and identifying partnerships, have moved us to what those partnerships can do and set us up beautifully for our next panel, which is once the partnership has developed and created something of value, where do we go from here? So thank you again to our panelists.